Good morning again, everybody. Welcome back. We can't slow down. We got to the interesting talks now. So I would like we're going to continue talking on the quality of genomic prediction. Several interesting talks, and we're going to start with one from Fred van Eeuwijk from Wageningen University. Thank you, Fred. Please, let's try and keep to the time because we are late, and we can't go indefinitely over, or we'll have a riot on our hands at lunchtime.
Okay, thank you, Graham. Yes, I mean, I hoped that Jeff was joking when he said that we would make up for the time lost in the previous session. Okay, I will not so much be talking about the quality of genomic prediction, but let's say I will be putting it a little bit into context, and let's say we will be comparing it with more classical GxE and QTL analysis, and the statement will be in the end that, let's say, there is a big overlap between GxE, QTL and genomic prediction for, let's say, the situation of multiple environments. So this is more or less what we're going to talk about. First, a little bit of a wrap-up of a number of concepts. Then, in some detail, a comparison between the philosophy underlying QTL mapping and genomic prediction. Then we will look at a multi-environment trial for the classical Steptomorax population, and we will try to find out whether the structuring of the environments, patterns in the environment, can be used to understand a little bit when genomic prediction will work and when not. To go one step further, we will then look at a simulated data set in which we know how the environments are structured given certain environmental gradients. And then, let's say, we'll go to the conclusions, and then probably is no time left, but if there's a lot of time left, then I could go on a little bit on something we did in crop growth modeling. So, some relevant literature, and I think Pancho talked especially about this one over here, I think which is a very nice paper, quite influential, and I immediately admit, let's say, that we took this paper, compared with a number of the other papers, and there's still something out of it that will be quite close to it. So, going back to yesterday, we were looking at the fourth and fifth dimension of phenotypic prediction, and that was by Yunbi Xu. I mean, we're not going to the fifth dimension, but to some extent what we'll be trying to do is we're trying to get closer to, let's say, how we think that the phenotype is driven by a vector of genotypic inputs, a vector of environmental characterizations, some functions on those genotypes and environments, and of course an integration over time taking into account developments. I mean, this looks from systems biology point of view looks quite nice, but actually when you look at the bottom of the slide you see what we usually are doing, and there is a rather big discrepancy between how biologists think that the phenotype is evolving from genetic and environmental information and how we do it in quantitative genetics. And in spite of the nice comparisons between me and Oscar Kempton and then what is it, Ronald Fisher by Pancho, I mean, I certainly don't think that, first of all, I can be compared with those guys, and second, there's still a lot of work to do on these infinitesimal models, so let's see whether we can bridge that gap a little bit. Now, this is mainly a colorful slide, and you will be happy to hear that I won't discuss the models on it. Mainly what I want to show here is that classically when we're analyzing GxE data and want to understand a little bit about what's going on, what we're really trying to do is we try to decompose our analysis of variance GxE term, which is here at the end actually in the two-way ANOVA form. This GxE term, we try to write it as something that depends on parameters that are either exclusively dependent on the genotype or on the environment. And you can see that, for instance, in this EMI model, in which here we have genotypic scores times environmental scores. We have it in the GxE model, which is a little bit the same, but let's say the difference with EMI is that there is no genotypic main effect. We have it in the so-called factorial regression models, in which we, let's say, regress the responses of the genotypes on some kind of environmental characterization Z, which could be temperature, humidity, drought, stress, whatever, and then, let's say, we estimate those betas, which are the sensitivities to such a stress. And actually, also, this model that we work with a lot, which is a model that includes QTL main effects and QTL by environment interaction, in the sense that this is the QTL effect, which is specific for each environment. So each environment is allowed to have a specific QTL effect for a given QTL locus. And you can see that also in this model we have, well, this genetic information which depends on the marker and the map, and we have the QTL effect, well, which depends in this case exclusively on the environment. So that is what we're trying to do when we model G/E, and I say a little bit more about the mixed model that we use for QTL/E. Actually, we derive it from the classical two-way analysis of variance model, and it simply is, let's say, partitioning the genetic main effect in something that can be explained by consistent or constitutive QTLs. Constitutive QTL effect, as Roberto would call it. It's not completely like that, but let's say this is the QTL main effect. And the G/E is partitioned equally so in, let's say, the deviations of, let's say, for a particular environment of the QTL main effect. Now it's easier to write these models in this way over here, where we have the expectation for the genotype I in the environment J, and this is an environmental intercept, let's say the mean for that environment, and then the rest is described by QTLs that are allowed to depend on the environment. Now the critical thing in modeling, let's say, those QTL/E models is that you need to take care of the polygenic residual effects. These genetical effects that are not accounted for by the QTL main effects or by the QTL/E interactions, these need to be modeled well, because otherwise we will get the same situation as we have in association mapping. We will get too many false positive QTLs. So it pays off, let's say, to find out whether the genetic variance in each environment has a proper value, heterogeneity of genetic variance, and whether the correlations between environments also are proper, so that each pair of environments has its own specific correlation. And later on, when we go to genomic prediction, we will see that actually we will be looking at the same kind of matrix, and Pancho already discussed that to some extent. So a little bit more about our classical QTL/E model. We're still talking about, in this case, biparental populations, but actually it can also be generalized to association panels and multiparental populations. I think So the main thing to take into account is, okay, I discussed these terms over here. This is the QTL part, and what we spend a lot of time on is trying to find out the structure of these residual genetic variations. And actually, what we do, and usually we don't write it explicitly so, but also Pancho spent some time on writing the variance covariances between, let's say, the genotypes and environments. When we are analyzing a biparental population in QTL mapping, we simply assume that all the genotypes are unrelated. This is not completely true, but for the analysis it doesn't matter so much. So for that reason, the variance covariance matrix is written as a product of an identity matrix, which indicates we assume the genotypes are independent, times, and that's important, times something that describes the relationships between the environments. So that's, is there genetic correlation between the environments, in this case on top of the QTLs. If we go later to genomic prediction, we will see that we forget about the individual QTLs, and we will be looking at the joint genomic prediction, the correlation between the genomic predictions in the different environments. And we will be writing the total variance covariance matrix in a similar way. Now, in a number of papers that, well, Pancho, myself, and other people, Hans-Peter Pifo, looked at, we spent time on finding, let's say, an adequate and parsimonious structure for the genetic covariances between the environments in the form of so-called factor analytic models, and these will come back also in genomic prediction. Now, just to give you a little bit of a flavor, and this is also the kind of classes that, let's say, we are giving to students in the human capacity building of the integrated breeding platform. This is an exploratory G/E analysis. We look at, well, the box plots for the different environments. There are 10 environments in this case, to check on the heterogeneity of genetic variance. We look at the ME and what is it, the GxE and the ME biplots, mainly to get a feeling for the genetic correlations between the environments and the heterogeneity of variance. And the next stage, if we decide that a particular covariance matrix is okay, we do a multi-environment QTL mapping. And this is the kind of stuff that, let's say, this year we will be teaching also in these integrated breeding platform courses. And this will be also an integrated breeding platform in the June version. So here you can see that, well, this is the overall test for QTL plus QTL/E on the 7 chromosomes of barley. And here down, you can see for each of the individual environments whether, let's say, the first or the second parent is contributing the allele that increases yield or a particular trait. And blue is, well, first parent, I thought, and yellow, red is second parent. And for instance, over here you can see that we have a crossover interaction, because in some environments it's the blue parent, in other environments it's the yellow-red parent. Okay, so that is QTL mapping. Now, maybe few people do it, but actually there's no reason why you would not do prediction with a QTL model as well, especially, let's say, when you spend time on developing a multi-QTL model. And actually the standard software in BreedingView and Gensted produces now these outputs, which tell you, let's say, what the fit is of the multi-QTL model, and it tells you what, let's say, the high value, which parent is contributing the high value allele, and also shows you which are the interesting genotypes to take for further crossing. And actually this is just like genomic prediction. So in a schedule you would, let's say, could describe it like this. We have the full set of phenotypes, typically in QTL mapping, multiple environments, and then we select a number of markers that are going to do the job. In genomic prediction, it's not that different, except for that we don't spend too much time on finding out where the QTLs are. We just simply take everything. And well, it appears as if we have a little bit more complexity in defining how well we're doing by splitting up, let's say, our genotypes in a training set and a validation set. But exactly the same schedule, of course, you could use in your multi-QTL studies. Or in the classical QTL studies, I mean, no reason why you would not work with cross-validation over there. So, if you look at the genomic prediction model, you will see that it's exactly the same as the QTL model, except in the QTL model we write a big Q to indicate that we select a number of markers, and here we write big M to indicate that we don't select anything. Because we are not selecting anything anymore, we still need to do something about our estimation problem. And Pancho said, well, this is a P larger than N problem, that is, we have more predictors, more markers than observations, than genotypes. And Pancho said it's a statistical problem. To be honest, I don't think it's exclusively a statistical problem, because we have to choose, we have to say something about how we are going, let's say, to restrict the parameter values, the estimates, the values that the estimates can take. And to do that, well, we have all kinds of methods, like the rich penalty, the less soup penalty, we have all these Bayesian methods. But in the end, the choice depends a little bit on the genetic architecture of your trait. So the more you know about the biology, the more efficient you can be in the choice of that regularization. Okay, now this is the, I think, essential slide in my presentation. And it's also very closely related to the central slide in Pancho's presentation. And what we're trying to do here is, I just explained to you that in genomic prediction across multiple environments, we have this term over here that indicates, well, these are our X's are related to the markers, and we are using all markers, and these A's are, let's say, related to the FX that are different, let's say, for each environment. And to model that efficiently in a mixed model context or in a Bayesian context, we would go back to something that looks like the original two-way analysis of variance model. Now be aware that I haven't included the genetic main effect, and this is also a difference with what Pancho showed. So here, this G/E term is including the genetic main effect plus the G/E simultaneously, and we're modeling that together. And we don't distinguish between the main effect and the G/E. Now, if you do that, the task in genomic prediction is that we have to find an efficient way of describing the variance-covariance matrix for this term, the genetic main effect and the G/E. And actually it comes down to writing the total variance-covariance matrix as something that depends on the genotypes and something that depends on the environment. And this is the separability constraint again that we also had in the initial slide when I showed the different models for modeling G/E. And what we do is, well, for the genotypes, relation between the genotypes in association mapping and also in genomic prediction, we base ourselves on the markers, we can base ourselves on the pedigrees, we can base ourselves on a weighted average of those two. We can also smooth these kinship matrices in some way. For the environments, you can do a number of things. And the thing that Pancho showed was, let's say, use environmental covariates to define something that is equivalent to the genotypic kinship. So Pancho defined an environmental kinship matrix and included that. But there's no reason why you would not use the same kind of modeling as we did before in the QTL by E analysis, let's say, and use vector analytic models or unstructured models that represent the relationships between the different environments. And actually, if to some extent you know your environments well and you have a good environmental characterization, you can group your environments in mega environments or in regions, and also that information can be used to find an official representation for this matrix over here, the part that describes the relationships between the environments. And we would call this thing GxE blub. Okay, let's see how this works. This is a simple situation, 5 locations, 2 years each. So first we will see in a moment, we will try to predict within 1 year and later between the 2 years. This is a Streptomorix population. This is also related to what Pancho showed. 150 genotypes, let's say, for training our model, then 50 left within year 1, let's say, to find out how well we're doing, validation. And if we go from year 1 to year 2, well, then actually we're trying to predict for the situation in which we didn't have environmental information. And here, for this block over here, we're going from genotypes that were not observed, and, no, this is from genotypes that were observed in environments that were observed, to here, genotypes and environments that were not observed, the most difficult situation that Pancho was talking about as well. Okay, so these are the 2 models once again. On the left, the classical G-blub model. On the right, the GxE blub, which as an extra feature has that it tries to model the relationship between the environments in one of the different ways I just explained. And, well, first you can do the GxE analysis, and once again this is to get you trained in finding out whether some of the environments are not very different. And here you see immediately this is the correlation matrix. You see that this 4th environment, which is a trial in Scotland that was done in 2003, that was added to the classical American Streptomorix trials. This Scottish trial is very different. It has low correlation with everything else, which probably will mean that for that environment I expect that the GxE blub will do a lot better than the G blub. Okay, we also check whether our training and validation genotypes are, let's say, covering the same space, something that always should be done in terms of quality control. This is, well, let's have a look how well we did. Well, I think the good news is that you can immediately tell that for those 5 environments that we were looking at, well, we will do always better if we use the GxE blub than we're using the G blub. So including the GxE, in this case, by using a factor analytic model, a standard factor analytic model we would also use in the QTL mapping, pays off. If you look at Scotland, the gain for Scotland is highest, and that is because Scotland was very outlying from the other environments, so it's not so strange that, let's say, for Scotland, the inclusion of the GxE in the genomic prediction will work out best. And if we look at the tested and untested genotypes, well, we can see that, yes, I mean, we do very well, let's say, when we predict for the genotypes on which we build a model, but we're still quite okay for the genotypes in which we are, let's say, going from the test set to the validation set, or from the training set to the validation set. And I have to say that the biggest difference, the difference is not so large anymore in this case, but the biggest difference once again appears in Scotland, which was very deviating from the rest. And the other one, which had a decent difference, is Wageningen, which also had more GxE than the others. Okay, now this is nice. My question was, I can do this with genomic prediction. Can't I just do it with QTL mapping as well? Well, these are, let's say, 2 examples of well, if you do genomic prediction, you usually take something between 10 and 100 subsets of the data to train your model. Here are 2 subsets to train the QTL model. And actually from this QTL model you can also immediately tell that Scotland is very different. And from the QTL model you can also find out what the genetic basis is for the correlations between the environments. So when I look at the performance in terms of prediction, well, actually the story is that the QTL by E does about as well as the genomic prediction in this case. Okay, so now we make it a little bit more complicated. We go to year 2, for which we don't have any phenotypic information, and especially this one over here in the corner is a very difficult one because this is genotypes that were not observed and environments that were not observed. And when we do that, well, it's not as fantastic as in year 1. And actually what you see now is that for a number of the trials in year 2, on the same locations, we see that actually the main effect blub does better than the GxE. And this is the case for those environments, let's say, that more or less present an average environment. If you're an average environment, probably you can better work with the average blub, the main effect blub, than with the specific GxE. In contrast, with the Scottish trial, it always pays off to use the GxE blub. And I also should now admit that, let's say, for some trials, we had a terrible prediction. But if you look at the correlation matrix, and we're not going to do in detail, if you look at the correlation from one year to the next, you can also identify that whatever you're going to do, whether you're using a G-blub or a GxE blub, you will never be able to predict the next year. And this, I think, is a central issue. I don't think that we should have as a criterion how well can we predict the next year. I think you need to identify what is your target population of environments, and you need to be able to predict to your target population from a training set that is part of your target population. You cannot predict one environment from the next when these environments don't have anything to do with each other. Exactly in the same way when it's hard to predict genotypes from genotypes that also are not related. Okay, now we go one step further. The same double haploid population, and we simulate 25 QTLs, and now we're going to make our QTLs dependent on temperature. And why are we going to do this? Well, we want to get a little bit close to the biology, and we also have good reasons that many QTLs are related to temperature. And actually what we do is we cross a red and a blue parent with each other that have these reaction norms, and this is temperature here, and this is yield. And you see that there is a green one that would be the best genotype possible if you combine the positive alleles from the two parents. And well, the way in which we parameterize the model is that it's a kind of curvilinear relationship. There's an intercept term that indicates the maximum yield that can be obtained. And then, let's say, the further you're away from your optimum temperature, the stronger the penalty. And if you get too far away from your optimal penalty, well, your yield will go down close to zero. That is more or less the model. This is the biological model. I mean, this is the biological model we made up. And these parameters over here, the intercept and, let's say, this stability thing, which more or less defines how quickly, let's say, the leveling off occurs, and this optimum temperature where the curve is located, those three parameters are driven by 25 underlying QTLs. That is the biological model. However, of course, we don't know that this is the case. So we analyze it by our classical QTL by E model or by our G/E blub. And well, why do we think it's reasonable to look at these reaction norms? Well, I say in stuff that we did together with Pioneer a number of years ago, and also in stuff we did together with Ignacio Romagosa and Gustavo Slaver, and we do find those linear dependencies of the QTL effects on the environments. So we estimate, we defined 10 environments, and this is the average, this is, let's say, the dependence of the average of the environment on temperature, and the environment 1 and 8 are cold environments, and 3, 2, and 6 to some extent are warm environments, and here we have a number of intermediate environments close to the optimum temperature. First thing to notice is that this is the graph for the genetic variance. The genetic variance is smallest in the best environments over here, and is largest, let's say, in well, the rather deprived environments. Okay, so once again, don't have time to explain all of this, but what we're trying to do here is this is the correlation matrix as a function of temperature. And on the diagonal, of course, you are well in the environment itself, and when you move off diagonal, there's a bigger difference in temperature between your environments. And the blue indicates that there is almost negative correlation between the environments, and green 0, and let's say yellow is about positive correlations. So, well, you can do the classical stuff to investigate what is going on with G/E, and the only thing I want to indicate here is that we can immediately tell that the environments 1 and 8 are very different, but we could have told already because 1 and 8 are quite cold environments, and when you look at the correlation structure, this is environment 1 with all the others. The only environment with 1 has a correlation, positive correlation, is environment 8. So those are the two cold environments. Actually, what's happening is you have cold environments over here in the GxE plot. You have warm environments over here, and here you have intermediate environments. And if you're now going to use your genomic prediction, well, you see that typically the G/E does quite good, actually exceptionally good. The main effect predictions are a lot worse, and they are especially a lot worse for the environment 1 and 8, because 1 and 8 are very cold environments that are rather atypical. For the environments that are closer to average, there is not that much difference between using the G blub and the G/E blub. If you use the QTL by E analyses, you will find out that in this case, your QTL by E predictions, this is the accuracies coming from the QTL by E analyses, is about the same as the genomic prediction. I mean, yes, it's lower, and it's sometimes substantially lower, but still it's quite high. So also taken as a base, if you take your mixed model QTL model as a baseline for genomic prediction, you're actually doing quite well already. Okay, we go one step further, still have 5 minutes, I can even make it. Now I'm going to do something that we have done in a European project as well, and I won't have time to talk about a European project, but actually in that European project we used a crop growth model to dissect yields in a number of parameters. And you can think of it in the same way as in the simulation example, because we can also tell, I mean, we can just pretend that we have found out that temperature is the driving force for our yield. And we simply fit this quadratic regression model to each of our genotypic reaction norms. We have these 10 evaluations for each genotype across the 10 environments. We have the corresponding average temperatures, and we just try to estimate an intercept, a slope, and a curvilinear parameter for each genotype. And then what we're trying to do is we're trying to use genomic prediction to fit values to those intercepts, slopes, and curvilinearities. So we're using this functional form for yields, but let's say we're using the genomic predictions for the parameters in those regression models to get to our predictions for yields. And well, here are a few examples of a few realized reaction norms. When we look at genomic predictions, actually this works quite well. It works almost as good as the G by E blub and the G blub. But we have to admit that there are a few environments, and especially environment 8, for which we actually don't understand what's going on. We are extremely badly performing for environment 8, but for most of the other ones we're doing quite well. Okay, so when I go to my final 3 slides, so what have I been trying to tell you? Well, first of all, I don't think that there's a very big difference between the genomic prediction and the QTL mapping. As long as you're using a powerful QTL mapping method, which means multiple environments, multiple traits. And the relationship between the environments will help you to understand whether you can or cannot expect a good prediction from either a G blub or a G by E blub. And especially when you have very deviating environments, then it will pay off to use a G by E blub, which, as I showed you, is in essence not that different from the multi-QTL models that we're using for our multiple trials. Something that I should say is, I think I talked about prediction on purpose, because I want to distinguish very clearly, and in a sense also Gary Edlin did so, I want to distinguish very clearly prediction from selection. And because if I want to predict for particular environmental conditions, I need to know what kind of target population of environments I'm looking at. I also think whether I'm going to use other trials to predict a particular environment. That kind of problem is an index selection problem. And sometimes you should not include environments that are very deviant from the environment that you're targeting. And let's say to find out what is going on in your specific set of trials, all those exploratory tools that I showed you I think are quite useful. And so, well, how did it look like? What is there to be done statistically? Well, we need to model the relationship between the environments, and we can do that by unstructured and factor analytic models, just as we are used to do in our classical QTL models for multiple environments. But I think a very attractive alternative is the one that Pancho showed, in which you base a kind of environmental kinship on a set of environmental covariates, environmental characterizations. For the genotypes, we have to do the same thing. We can either use the pedigree, we can use the markers, or we can use a combination. And I also have to say that it pays off to try to find some grouping in your genotypes, because that can reduce the complexity of the genetic part of the variance covariance matrix, and that is certainly necessary when you want to combine some complexity on the genotypes with some complexity on the environments. And I also have to take, I assumed that I could separate the influence of the genotypes and the environments. I think more complicated things are possible, but we're not yet there. And so finally, I also think that one of the ways forward is an integration with, well, functional forms for yields, for complex traits, using crop growth models, and finding out what are the simple components that we can easily model, either with QTLs or with genomic prediction, then introduce those genomic predictions for those parameters back in the nonlinear crop growth models, and in that way, let's say, try to predict for new genotypes and new environments. And some of that stuff can be found already in this paper from 2010, in which, let's say, some of the stuff that especially I've been doing with Scott Chapman is available. And that's it, and it's 30 seconds left. It was too much.
Harry.
You were showing in one of your slides several scenarios where in some of the environment, say your QTL into E environment is as good as genomic predictions. Whereas when we are talking of both G and GxE, G was much better. Main effects were providing much better prediction than GxE. So what could be the driving force in these two scenarios?
Okay, let me see what I understood, because your question is, sometimes the G blub did better than the GE blub?
Yeah.
And how is this possible?
No, I mean, what could be driving in those environment this kind of predictions than in other environments where you get GxE better than G?
No, I mean, to be honest, what will be doing better depends to an enormous extent on the set of environments that you're analyzing. Because if you have a very deviating environment from, let's say, well, the majority of your trials, then this deviating environment will force you to use a GxE blub. If you're looking at the number of trials that show some consistency and homogeneity, then quite often you're better off by using the G blub for that set of homogeneous trials. And that also emphasizes, I think, a very strong point in terms of structuring your environments. I think it always pays off to define your environments in a set of regions or subpopulations of target environments. And as soon as you have identified those, then actually you can simplify your prediction by going, let's say, for G blub or main effect blub or main effect QTLs. So I think first look at the GxE, structure of your environment, and try to make homogeneous groups. And if you do that, typically main effects will do quite well. If, however, you have a hodgepodge of all kinds of different trials, with some of the trials very deviating, you will pay the price by having to use more complicated models for prediction.
Why I did ask this was that when you have high GxE interaction in your analysis, then obviously your heritability estimates are very low in those.
That's not true.
And that's really for us a common sense that whenever your heritability estimates are low, your predictions would not be that precise and great.
No. This is definitely not true, because I give you the example. Let's say some of the data sets that we analyze, and also goes back to the work that Shell Marcel did. If you have a well-watered trial together with 10 extremely dry trials, then that well-watered trial will stick out on all sides, but it's the environment with the highest heritability. So it's not necessarily so that the environment that sticks out is the one with the low heritability.
I think we're going to need to take this one over lunch. You had a question? One quick.
Or not, but so in traditional phenotypic breeding programs, you make selections from year to year, and you're sort of stuck with the phenotypes you get in any sort of year, no matter how aberrant that year is, right? But in genomic prediction, as you build data up across time, you have the option whether or not to include data from a certain year for making selections. So do you have any ideas on when to include years in training sets and when not to? Any sort of criteria to help us decide?
Well, let's say this is something we're looking at. There is a classical paper by Jacques Decker from 2007 in which he looks at combination of phenotypic selection, marker system selection, and genomic prediction. Actually, I had a few slides that mentioned that index selection would be the way forward. I mean, to be honest, I cannot give you a clear-cut advice on that, but it depends on the correlation of that particular year with the long range average of let's say, that environment across years. It's the how representative is that particular year of general conditions in that environment. Yeah, this is not a fantastic answer, but
we're into the very hard discussions now, but we'll have to take them off to lunch. Thank you, Fred, and I would like to ask John Hickey